Welcome to a short presentation of the CABS Flex 3.0 server. This web-based tool allows protein flexibility modeling and peptide structure prediction. CABS Flex 3.0 works in two modes: flexibility modeling and peptide modeling. Let's start with flexibility modeling. This mode simulates structural flexibility in proteins. You'll find basic options here and advanced options here. One important setting is the flexibility mode. There are 4 options, each reflecting a different level of flexibility. For most users, we recommend the flexible mode. All settings are explained in the How-To section. To start a job, just provide a PDB structure and click Run. Let's run an example using structure 8 DoD. Click Run, and the job is submitted. Submitted jobs can be found under the Queue tab. Here's a finished job. A typical job takes from a few minutes to a few hours, depending on the size of the protein. Each job has a dedicated results page with 4 tabs: Project Information, Models, Contact Maps, and Fluctuation Plot. Under Project Information, you'll see job details, chosen parameters, and downloadable files. Now, to the Models section. The Structure tab shows predicted flexibility using line thickness and color. The legend shows fluctuation values in angstroms. You can change the color scheme to PLDDT, B-factor sequence, chains, secondary structure, or custom colors. Under the Models tab, we see the main CABS Flex prediction, 10 models showing the protein's flexibility, color-coded by domain. You can play an animation of these structures. The All Models tab shows all 10 models overlaid. On the right, you can show the initial structure, reset the view, or display disulfide bonds. The viewer supports full-screen mode. You can also export high-resolution images. In the Contact Maps tab, you'll find interaction maps from the simulation. Hover over the map to see contact frequencies. This protein consists of 3 chains, and you can view maps for individual chains or interchain contacts. Clicking a contact shows it in the 3D structure viewer. Now, the fluctuation plot. This shows predicted root mean square fluctuation values, aligned with secondary structure. Here, the highest fluctuations in chain H reach around to angstroms. You can switch between chains to view their flexibility profiles. Let's go back to the home page and explore peptide modeling. This mode requires only the peptide sequence. Secondary structure is optional. The server can predict it automatically. There are fewer options than in protein modeling. You can define disulfide bonds or make the peptides cyclic. Here's an example of a cyclic peptide job. Just like before, the results include 10 predicted structures. They're ranked from Model 1, the best score, to Model 10. You can view their structural diversity in the viewer. For more details, check the How-To tab. That's the overview of CABS Flex 3.0. It's a fast, user-friendly tool for protein and peptide modeling. Thanks for watching.